All right, so in this video, we are going to be going over chapter 6, which is titled A Tour of the Cell. And we know that all organisms are made up of cells, and that cells can be defined as the simplest collection of matter that can be alive. So studying cells is really important, and understanding them is what made possible with the invention of the microscope. So microscopes were first developed in the late 16th century, and major improvements to them came in the early 17th century. So in 1590, the light microscope was invented, right? So in 1590, the light microscope was invented. And it was invented by somebody named Zacharias Janssen. And then in the 1670s, another scientist named Antony van Leeuwenhoek greatly improved the microscope lenses. And Leeuwenhoek's work, alongside some other biologists and scientists, it laid the foundation down for cytology, right? And cytology is just the study of cells. So light microscopes, they're also known as optical microscopes, right? And light microscopes work by using their visible light to magnify onto specimens. And these light microscopes, they allowed scientists to observe individual cells, such as bacteria, plant, and animal cells. But they do have limited resolution, which makes it unable to reveal the internal organelles of the cell. And we have key properties of microscopes, which include magnification. And magnification, it is just the degree to which an image is going to be enlarged in comparison to the actual specimen. It's determined by our eyepiece lenses and also by our objective lenses. Resolution, it's another property that refers to the ability to distinguish two close objects as separate entities. And the higher the resolution, the clearer the details. And contrast is the last property that we're going to go over. It is the difference in light intensity between the specimen and the background. And we can actually improve contrast by staining and labeling. So we have different kinds of microscopes, and one of these microscopes, they're called electron microscopes. And our electron microscopes, they allow for a much higher resolution than our light microscopes. They are really essential for studying those fine details of cells and their organelles, and they actually work by using a beam of electrons and onto the specimen instead of using light. And we have two different kinds of electron microscopes. We have scanning electron microscopes, and they're actually going to produce 3D images. And they're going to provide these really detailed surface structures. And a downside of these microscopes is that we can only see the external structures, and the samples, they also have to be coated in metal, which is going to alter the cell. And the next kind of electron microscope that we have is a transmission electron microscope. The electrons, they're going to pass through this really thin specimen, and that is going to be able to reveal our internal structures in really high detail. But a disadvantage of it is that it's going to require complex sample preparation, because our specimens, they have to be extremely thin in order for the electrons to pass through them. So overall, both scanning and transmission electron microscopes, they're going to require our specimens to be placed inside of a vacuum. And doing that means that the living cells cannot be observed. It is going to require killing the cells to observe their structures. And even though this preparation of processing and preparing a cell can alter its structures, our electron microscopes have revolutionized cytology by being able to reveal the intricate details that are far beyond the capabilities of what light microscopes can show us. So cell fractionation, it is a laboratory technique that's actually going to break open a cell and separate their internal components based on size and density. Okay, so size and density. And this is going to allow for the isolation of specific organelles, and that's going to make it easier to study their structure and function individually. So the way that this works is that you use a homogenizer to break open cells. And once those cells break open, they're going to release content. Then we're going to spin that sample in a centrifuge at different speeds. And the heavier an organelle is, it's going to cause it to settle first at higher speeds. And when we have smaller organelles, we're going to have to separate them in later spins at lower speeds. But cell fractionation, it has led to really important discoveries. So for example, this method actually identified mitochondria as the site of cellular respiration. So eukaryotic cells have internal membranes that compartmentalize their functions, which is going to allow for the greater efficiency and complexity in their cellular processes. And our eukaryotic cells, they evolved from prokaryotic cells, and then they developed specialized structures known as membrane-bound organelles. So we do have differences between our eukaryotes and our prokaryotic cells. Our eukaryotes, they're going to belong to domain eukarya, and it's going to include our protists, fungi, animals, and plants. Our eukaryotes are larger and more complex, and they contain these so important membrane-bound organelles like the nucleus, mitochondria, and like the endoplasmic reticulum. Our prokaryotic cells, on the other hand, they're found in domain bacteria and archaea. They are smaller and they are more simple, and they lack membrane-bound organelles. So they don't have those membrane-bound organelles. And our prokaryotic cells, they're considered our ancestral cells because they appeared before eukaryotic cells in evolution. And eukaryotic cells, their internal membranes, that's what's going to allow them to perform specialized functions and make them more advanced than our prokaryotic cells. So regardless of the cell type, every single cell is going to share these four common structures. The plasma membrane. The plasma membrane is a selective barrier that is going to regulate the movement of substance in and out of the cell. Our cytosol, it is a semi-fluid substance that we find within the membrane, and that's where our subcellular components are going to be suspended. We're also going to find chromosomes, and we know that chromosomes are the structures that carry genetic information in the form of DNA, and we also know that DNA is going to direct protein synthesis and other cellular processes. So we're also going to find ribosomes in all of our cells. And this is because ribosomes are the site of protein synthesis. They are essential for producing the proteins that are needed for cell survival, whether it's eukaryotes or prokaryotes. So all four of these features, they're going to be present in both of our prokaryotic and eukaryotic cells, and it's going to form the foundation of all life. So now that we covered the similarities between the cells, I want to point out their differences in more details. So our prokaryotes, they do not have a nucleus. The DNA is going to be located in a region called the nucleoid instead of being inside of a membrane-bound nucleus. Our prokaryotes, they do not have membrane-bound organelles. Their cellular functions, it occurs directly inside of the cytoplasm. And when we're talking about prokaryotes, the cytoplasm is referring to the entire space within the plasma membrane. And our prokaryotes, they're smaller. They're typically 1 to 5 micrometers in diameter. On the other hand, we have eukaryotic cells. And our eukaryotic cells, they have a nucleus. So they contain a nucleus that is a double membrane structure that is going to house DNA. And our eukaryotes, they also have these membrane-bound organelles. Organelles like mitochondria and the endoplasmic reticulum, which are going to compartmentalize cellular functions. And when we're talking about the cytoplasm inside of eukaryotic cells, it's referring to the region that we have between the plasma membrane and the nucleus. And our eukaryotes, they're larger than our bacteria. They're about 10 to 100 micrometers in diameter. So because we have larger eukaryotic cells, those cells, they had to evolve, they had to adapt in order to maintain efficiency. The size of a cell is limited by its ability to carry out cellular metabolism efficiently. So as our eukaryotic cells grew in complexity, those membrane-bound organelles, they developed to help distribute tasks and allowed for the cell to function properly despite its larger size. So the plasma membrane of a cell, it is a selective barrier that is going to allow for the regulation of molecular passage in and out of the cell. It's going to regulate the entry and exit of oxygen, of nutrients, and waste for the entire cell volume. And that's why our cells are limited to a specific size. The surface area and volume of a cell, they must retain a certain relationship in order for that surface area to be able to keep up with the demands of the cell. And the problem with increasingly larger cells is that the volume is always going to grow faster than its surface area. And that just means that because we have less surface area and more volume, the plasma membrane, it cannot keep up with the metabolic requirements of that cell. So when we have smaller cells, we're going to have a higher surface area to volume ratio, which is going to allow for more efficient chemical exchange. But when we have larger cells, there's going to be a lower ratio of surface area to volume. And that's going to cause for there to be slower and less efficient exchange. And when we're looking at this figure, what it's showing us is just showing us a small and simplified snippet of what the plasma membrane is like. It's a phospholipid bilayer that contains important proteins like peripheral proteins and like our integral proteins that are going to allow for the passage of important molecules through the membrane for proper function. So because our eukaryotes diverged from prokaryotes, originally they did not have membrane-bound organelles. But as they grew in size, they became challenged because their large size made them less efficient because of their inappropriate surface area to volume ratio. So our eukaryotes, they adapted in order to be able to efficiently maintain all of their metabolic processes. So one of the adaptations that occurred was the formation of accessory structures, which accessory structures, they're going to project off of cells, and the way that they work is by increasing that surface area that a cell has. So an example of an accessory structure is going to be microvilli, and we can find these microvilli in intestinal cells where there is this really high rate of absorption. So having more surface area will be beneficial to the cell so that it can absorb more nutrients into it. And another adaptation that they developed is compartmentalization. And what this means is that our eukaryotes, they started to compartmentalize their internal membranes, which then divided the cell's organelles into separate membranes. And these membrane-bound organelles, they allowed for the more efficient metabolism because it allowed these cells to do a variety of metabolic processes simultaneously. Because normally, so many metabolic processes, they can't happen at the same time, which is what we see in our prokaryotes and why they're so simple. But because we have separated membrane-bound organelles, we can do a large variety of metabolism at the same time. And it's important that we know that every single organelle is going to be surrounded by their own specific membranes. So for example, the nucleus of a eukaryotic cell, it's surrounded by a double membrane, which is called the nuclear envelope. And each side of the nuclear envelope is going to have distinct functions, and it's going to be connected to other organelles by direct or indirect manners. Another example is our mitochondria, and our mitochondria also has a double membrane. And that double membrane, it has distinct functions depending on the side of the membrane that we're looking at. And the endoplasmic reticulum is also another example. It's surrounded by a single membrane, but that single membrane can then be divided into a smooth and rough portion, and both portions have different functions. But overall, the compartmentalization of subcellular components, it allowed eukaryotes to become more efficient. So the nucleus, it is a defining feature of eukaryotic cells. It acts like a control center, which is where genetic instructions are going to be stored and used. The nucleus, it has a variety of functions which include genetic storage. So the nucleus, it contains most of the cell's DNA, which is going to carry those genetic instructions. But we actually have two other structures that DNA can be found in. So DNA, it can also be found in mitochondria and in chloroplasts. Chloroplasts are only plant cells. Meanwhile, our mitochondria, they're found in animal and plant cells. So our nucleus, it's also going to control gene expression. The nucleus, it's going to regulate cellular activities such as growth and division, and it's going to do this by controlling the genes that are being expressed. And another function of the nucleus is protein synthesis. And the nucleus, it's going to direct protein production by indirectly synthesizing messenger RNA. Indirectly. So mRNA, it's going to leave the nucleus, and it's going to then be translated into proteins by ribosomes inside of the cytoplasm. So the nucleus, it's enclosed by a double membrane called the nuclear envelope. And the nuclear envelope has an outer portion that faces the cytoplasm. We call that the cytoplasmic envelope. And the key structures of our nuclear envelope is that it's going to contain these pores all along it. And through these pores, it's going to allow materials to pass in and out of the nucleus. But these pores, they're lined by something called the pore complex. And the pore complex, it's a protein structure. And that protein structure is actually what's going to regulate what enters and exits the nucleus. And the inner portion of the nuclear envelope, it's called the nuclear lamina. It's going to line the inner side of the nucleus, this side, the side that faces the nucleus. And the inner portion of the nuclear envelope, it's going to be supported by the nuclear lamina. And it's a network of proteins that's going to provide structural support and help maintain the shape of the nucleus. And we also have something called a nuclear matrix. This is hypothetical at this moment. And it's hypothetical because we have some evidence to suggest the existence of a nuclear matrix, which is a fibrous framework that can help organize nuclear activities, but we have not confirmed its existence yet. But together, these structures of our nuclear envelope is going to not only help protect the nucleus, but it's going to regulate the transport and maintain nuclear integrity. So inside of the nucleus, we have several key structures that help organize and regulate genetic material. These include our chromosomes, our chromatin, and the nucleolus. So our chromosomes, they're going to carry genetic information in the form of DNA. But when we're inside of the nucleus, we're going to actually find our chromosomes as chromatin. And chromatin is a long DNA molecule that has many proteins. So our chromatin, it's actually a complex. And what I mean by it's a complex is that it's DNA and proteins together. And our chromatin, it's going to help with gene regulation and packaging. Another structure that we have inside of the nucleus is the nucleolus. And the nucleolus, it's this really dense and fibrous region that we have inside of the nucleus. It is the site of ribosomal RNA synthesis, rRNA. It's where this rRNA is going to be synthesized, and it's where it's going to be assembled. And once assembled, we're going to get ribosomal subunits that are going to exit the nucleus through the pores. And they're going to participate in protein synthesis inside of the cytoplasm. So these interior structures, they're going to ensure that genetic material is stored, it's organized, and processed efficiently within the nucleus. So the nucleus, it plays an extremely important role in synthesizing rRNA and messenger RNA, mRNA. And these two kinds of RNA, they are essential to protein synthesis. So we already discussed that rRNA is assembled and synthesized inside of the nucleus. So once our rRNA is formed, it's going to move into the nucleoplasm, which is just this space here inside of the nucleus. And once it moves into the nucleoplasm, it's going to interact with cytoplasmic proteins that were able to enter through these nuclear pores. And what's going to happen is that it's going to form a combination of two ribosomal subunits. It's going to form a large and small subunit. And those subunits, they're going to exit out of our cytoplasm through our nuclear pores. And later, those ribosomes, they're going to combine and they're going to form functional ribosomes when those two subunits come together. So mRNA, it's also synthesized inside of the nucleus, and it's going to carry genetic instructions that are necessary for protein production. Our mRNA is also going to exit through nuclear pores into the cytoplasm, and when we're in the cytoplasm, ribosomes that are already completely assembled, they're going to bind to mRNA, which is when translation is going to begin. Our mRNA is going to thread itself through the ribosome, and that ribosome is going to read those instructions that are found within the mRNA. And then the ribosome, it's going to assemble polypeptides, which is just forming proteins based on mRNA sequence. And if we are going to remember, polypeptide chains, they're the first level of protein structure. So when we're looking at the figure, what we see is that we have in the nucleus, we have chromatin, right? So here we have chromatin. And this DNA, it's going to synthesize our mRNA. And that mRNA is going to carry the genetic code for proteins. And it's going to exit from the nucleus into the cytoplasm through those nuclear pores. And when we're looking at the cytoplasm, we already have our ribosomes assembled. So once our mRNA enters the cytoplasm, it's going to thread itself through the ribosomes like this. And once it threads itself, it's going to, the ribosomes, they're going to translate that mRNA into polypeptide chains, which are the first level of protein structure. So we've established that ribosomes are the site of protein synthesis, and we can actually find ribosomes in two locations. We can either find them in the cytosol as free ribosomes, or we can find them lining the endoplasmic reticulum of the nuclear envelope. And these that are lining the endoplasmic reticulum, they're known as bound ribosomes. And both of these forms of ribosomes, they have different functions. Our free ribosomes, they're going to synthesize proteins that are going to function in the cytosol of the cell. And our bound ribosomes, they're just going to synthesize proteins for membranes, for organelles, and secretion. So what happens when we look at the figure is that we see a small subunit of the ribosome and a large subunit of the ribosome. And they're going to come together, and mRNA, which is a single-stranded molecule, threads itself through this assembled ribosome. And when we're looking into the large ribosomal unit, we're going to find tRNA. And tRNA stands for transfer RNA. In this transfer RNA, it's going to read this mRNA sequence, and it's going to bring the correct amino acids to the growing polypeptide chain. And these amino acids, they're going to link through peptide bonds. And when they link, that's when we get that forming of a polypeptide chain. And this process, it's known as translation. Translation is the conversion of genetic information into proteins. So we have something called the endomembrane system, which is really important to us. The endomembrane system, it is a network of interconnected membranes, and this is found inside of eukaryotic cells. And it consists of 6 key structures. It consists of the nuclear envelope, which surrounds the nucleus and regulates material exchange. It consists of the endoplasmic reticulum, which is going to synthesize proteins at its rough endoplasmic reticulum, and it's going to synthesize lipids at the smooth endoplasmic reticulum. It consists of the Golgi apparatus, and the Golgi apparatus is going to modify, sort, and ship proteins into lipids. It has lysosomes, and our lysosomes, they're going to contain digestive enzymes for breaking down macromolecules. It also is going to contain vacuoles, which are going to store nutrients, waste, and other materials. And the last component of the endomembrane system is the plasma membrane, which is going to control what enters and exits the cell. So the overall functions of the endomembrane system is to transport materials between organelles. It's to synthesize molecules, including proteins and lipids, and it's important in the maintenance of cellular structure by compartmentalizing different functions. And the components of the system, they're going to be connected either by a direct physical contact or by an indirect mechanism, which is going to be through vesicular transport. But this is just going to ensure that we have efficient protein processing and cellular metabolism. So the components of the endomembrane system, they are connected through 2 main mechanisms. They have a direct connection, which is a continuous membrane, which includes our nuclear envelope and our rough endoplasmic reticulum, which is what we see here. Right here, we have our nuclear envelope all around here, and we see that it's directly connected to the lumen of our rough endoplasmic reticulum, which is going to form one single structure. And we also have a kind of direct connection between the endoplasmic reticulum and Golgi apparatus, but it's not a continuous membrane. What happens is that vesicles, they're going to bud off of the rough endoplasmic reticulum. So we see here a vesicle that's starting to bud off. And that vesicle, it's going to directly transport material to the Golgi apparatus. And this connection is considered direct because vesicles, they're carrying proteins and lipids that bud off of the endoplasmic reticulum, and it's being immediately and specifically sent to the Golgi apparatus, and it is not interacting with any other organelles first. So indirect mechanisms of connection include vesicular transport. And what that means is that we're going to have vesicles that are going to transport proteins and lipids from the endoplasmic reticulum to the Golgi apparatus, and from the Golgi to other organelles. Our lysosomes, they're going to receive enzymes from the endoplasmic reticulum and the Golgi apparatus. And then our vesicles, they're going from the Golgi and other organelles, and then they're going to fuse with the plasma membrane to deliver or release materials that we have outside of the cell. And these connections, they're going to ensure the efficient material transport and coordination of cellular functions. So the endoplasmic reticulum, it is directly connected to the nuclear envelope, which is what we see here. And it's going to play a really important role in molecular transport. It's going to make up more than half of the membranes in a eukaryotic cell, and it has two distinct regions. So it has a rough endoplasmic reticulum, which is going to be studded with ribosomes. And those ribosomes are going to be responsible for protein synthesis. So the functions of the rough endoplasmic reticulum is protein synthesis. It's going to produce proteins that are going to be secreted and then incorporated into membranes or sent to other organelles. It also functions in the folding and modification of proteins. It's going to ensure that newly made proteins are appropriately folded and modified. And then we have our smooth endoplasmic reticulum. And the smooth endoplasmic reticulum, it's smooth because it doesn't have any ribosomes on its surface. And its function is lipid synthesis and metabolic processes. It's involved in the detoxification of our cells. It's going to be involved in calcium storage and carbohydrate metabolism. But the endoplasmic reticulum as a whole, it's going to act as a transport network, which is going to direct proteins and lipids to their next destination inside of the endomembrane system. So the smooth endoplasmic reticulum, it is a enzyme-rich organelle that is involved in multiple metabolic processes. So unlike the rough endoplasmic reticulum, the smooth endoplasmic reticulum lacks ribosomes on its surface, and it's going to specialize in non-protein related functions. And that includes lipid synthesis, right? It's going to produce phospholipids, steroids, and oils. And all of these are essential for the cell membranes and for hormone production. It's going to have a role in carbohydrate metabolism. So it's going to help break down and synthesize carbohydrates for energy storage and release. It's also going to have a role in detoxification. So it's going to break down drugs and alcohol and toxins inside of our liver cells, which make harmful substances water soluble for excretion. So that's what our smooth endoplasmic reticulum does. It's going to allow those harmful substances to leave the cell. And the smooth endoplasmic reticulum, it also has a role in storage of calcium ions. So it's going to store our calcium ions, which is really important for muscle contraction and cell signaling. So I already mentioned that the smooth endoplasmic reticulum, it plays a major role in detoxification, but particularly in the liver cells. And when we think about it, when a person frequently consumes alcohol or drugs, our smooth endoplasmic reticulum, it's actually going to proliferate. And because we have more of our smooth endoplasmic reticulum, it's going to increase the ability to detoxify the liver. And that's actually going to lead to a drug tolerance. And that's what's going to cause larger doses to be needed in order to achieve the same effect. So our rough endoplasmic reticulum, it has a few different functions. It functions in secretory cells. So what happens is that we are going to have protein synthesis and folding. So our bound ribosomes, they are going to synthesize polypeptides, and then those polypeptides, they're going to enter the endoplasmic reticulum lumen. And those polypeptides, they're going to end up folding into their functional shape as they move throughout the rough endoplasmic reticulum. And many of those secretory polypeptides, they are glycoproteins. And glycoproteins, they are just proteins that have attached carbohydrates. It's also going to work in protein transport, okay? So we're going to have newly synthesized secretory proteins, and then those proteins are going to be packaged into vesicles, transport vesicles, and then those vesicles, they're going to bud off of the rough endoplasmic reticulum and travel to other destinations like the Golgi apparatus. It's also going to function in membrane production. Okay, so the rough endoplasmic reticulum, it's going to produce membrane-bound proteins that are going to integrate directly into the endoplasmic reticulum membrane. And it's also going to function in phospholipid synthesis. And the synthesis of phospholipids, it occurs from cytosolic precursors, which is going to expand that cell's membrane system. And since the rough endoplasmic reticulum, since it's so abundant in cells that secrete proteins, such as our pancreatic cells that are going to release a lot of digestive enzymes, it's actually going to play a crucial role in both protein processing and in membrane synthesis. So the Golgi apparatus, it has a lot of functions. So basically, those transport vesicles that budded off of our endoplasmic reticulum, they're going to deliver proteins and lipids to the Golgi apparatus for further processing. And once those vesicles are inside of the Golgi apparatus, the vesicular contents, they're going to be modified, sorted, and packaged for transport to their final destination. So the Golgi apparatus, it's actually going to be composed of flattened stacks that we call cisternae. And these cisterna, they have membranes that separate its internal space from the cytosol, right? So here's one of our flattened cisterna. And nowhere do the cisterna, the lumen of the cisterna and the cytoplasm, they don't mix. They're completely separated. So our Golgi apparatus is going to have a cis face, and that cis face is always going to be near the endoplasmic reticulum. And that cis face, it's going to receive materials by fusing with the transport vesicles from the endoplasmic reticulum, such as this one. And then the Golgi is also going to have a trans face, and that trans face is always going to be away from the endoplasmic reticulum. It's going to have vesicles that are going to bud off of the Golgi, and those vesicles, they're going to contain modified molecules, and it's going to be sent to other parts of the cells. So modification of proteins and lipids, it occurred between the cis face and the trans face. So from when our proteins and lipids move from the cis to trans region, they undergo modifications that are going to be facilitated by specific enzymes that we find inside of the cisterna. So the Golgi apparatus, it's also going to play a really important role in modifying the carbohydrate portions of glycoproteins. And the Golgi apparatus, it also has the capability to synthesize its own macromolecules. But the final products, they're going to be secreted from the trans face of the Golgi inside of transport vesicles, and then these transport vesicles, eventually they're going to fuse with the plasma membrane or with other organelles. And the Golgi apparatus, it is a really dynamic structure. It's a dynamic structure that is going to manufacture and refine its products in a stepwise manner, with each cisterna containing a specialized set of enzymes for specific modification. So lysosomes, they are another component of the endomembrane system, and they are digestive compartments. Our lysosomes, they are membrane-bound sacs of hydrolytic enzymes that are going to enable animal cells to digest macromolecules. And our lysosomal enzymes, they work best when they're at an acidic pH. But our lysosomes, they can actually rupture. And you would think because they're acidic and they rupture, it's going to mess with the pH of our cell. But that's not true. It's not true because if we have a few lysosomes that rupture, their enzymes, they're going to be mostly inactive at the pH of the cytosol because cytosol has a neutral pH. But if we were to have like a massive lysosomal rupture, it can actually lead to something called autodigestion, cell autodigestion and destruction. So our lysosomal enzymes and membranes, they're going to be initially synthesized by the rough endoplasmic reticulum, and then they're going to be modified and processed inside of the Golgi. And some lysosomes, they're going to actually be formed by budding from the transphase of the Golgi. But it's important that we note that our lysosomal membrane proteins, they are protected from enzymatic digestion by their three-dimensional conformation, which is going to shield vulnerable bonds from hydrolysis. So our lysosomes, they are protected because they do not undergo hydrolysis. So our lysosomes, they have a variety of functions, right? So one of their functions is intracellular digestion. And what happens during intracellular digestion is that lysosomes are going to just digest foods that are in cells. So for example, when we're talking about amoebas or other unicellular organisms, they're going to feed by something called phagocytosis. Okay, so amoebas, they feed by phagocytosis. And phagocytosis is the engulfment of smaller organisms or food particles. So when they engulf food, they are going to have that ingested material in something that's called a food vacuole. And then that food vacuole, it's going to fuse with the lysosome for digestion. So as our macromolecules are broken down, their monomers, they're going to pass into the cytosol and serve as nutrients. But in humans, right, so in humans, we have something called macrophages. And macrophages, they are white blood cells. And those white blood cells, they use phagocytosis to destroy harmful bacteria and defend the body. So autophagy, it is the recycling of organelles and macromolecules. And our lysosomes, they're going to help in the recycling of the cell's organelles and macromolecules through a process, and that process is autophagy. So during autophagy, a damaged organelle or even cytoplasmic material, it's going to be enclosed inside of a double membrane. And that double membrane, it's going to fuse with the lysosome. And once it fuses, it's going to break down its contents and release organic monomers back into the cytosol for reuse. So when we're looking at this figure, what we see here is a double membrane vesicle, which is called an autophagosome. And it's going to begin to form around the targeted molecules or cellular debris. And this membrane, it's thought that to actually originate from the endoplasmic reticulum or from other membrane sources. So what happens is that when we have the completion of our autophagosome, it's going to engulf and fuse with a lysosome. So the damaged organelles and cellular material, they're going to be completely enclosed within the autophagosome, which is going to isolate them from the rest of the cytoplasm, and then we're going to get the fusion of the lysosome. And that is going to form an autolysosome. And the lysosome, it's going to contain hydrolytic enzymes that's going to degrade the enclosed material. So the cellular components, they're going to be broken down into basic building blocks like fatty acids, like sugars, and like amino acids. And then those monomers are going to be recycled back into the cytoplasm for reuse in new cellular processes. And that's because every single system, every single cycle inside of a cell, it is not wasted. One cycle's waste is fuel for another cycle. So everything is going to be recycled and used. So vacuoles, they are another part of the endomembrane system, and they have a really diverse function inside of our cells. So our vacuoles are membrane-bound organelles that are going to be found in both plants and animal cells. They are large vesicles that are derived from the endoplasmic reticulum and the Golgi apparatus. And it's going to have membrane-bound compartments that's going to have a variety of functions depending on the cell type. And some of these functions include the storage of nutrients and waste products. It's going to help in the maintenance of cell pressure. It's going to aid in intracellular digestion and the recycling of materials. And it's actually going to help with the maintenance of shell shape and homeostasis as well. So we have different types of vacuoles, and one of the kinds of vacuoles that we have are food vacuoles, and we are going to find them in some single-celled organisms such as amoebas, and we're also going to find them when a cell engulfs food particles via phagocytosis. And these food vacuoles, they fuse with lysosomes where enzymes are going to digest food. And another type of vacuole that we have are contractile vacuoles. And these contractile vacuoles, they are found in freshwater protists. So they're found in freshwater protists and some unicellular organisms. But what they do is they're going to pump excess water out of the cell to maintain an ion balance and osmotic stability. So we have vacuoles inside of our plants and fungi, and they're going to function in enzymatic hydrolysis that is going to be similar to lysosomes in animal cells, and they're going to aid in the storage and structural support in our cells, which leads us to our central vacuoles. Our central vacuoles, they are found in mature plant cells, and they are enclosed by a vacuolar membrane. And that membrane, it's going to selectively transport solutes, which is going to make that vacuolar fluid different from the cytosol. And the functions of these central vacuoles inside of our plants, it's to store proteins, ions, pigments, and metabolic byproducts. It's going to maintain turgor pressure, which is going to allow the plant to maintain rigidity. And it's going to facilitate growth. It's going to absorb water, and then the vacuole is going to expand without the need for any additional cytoplasm. So we have two structures that are called a mitochondria and chloroplast. And if you remember, aside from the nucleus, these two structures are the only ones that contain DNA. And they have an important function, and they both function to convert energy into forms that cells can use for work. The mitochondria, we can think of the mitochondria as the powerhouse of the cell. It's going to facilitate cellular respiration by using oxygen, and it's going to generate ATP from oxygen by breaking down sugars, fats, and other fuel sources. Chloroplasts, on the other hand, they're going to be found in our plant and algae. and they are the sites of photosynthesis. So our chloroplasts, they're going to capture solar energy and convert it into chemical energy by synthesizing organic molecules and such as sugars from carbon dioxide and water. So our mitochondria and chloroplasts, they have a theory that is going to suggest where they evolved from, and that's going to be called the endosymbiont theory. And it just suggests that our mitochondria and chloroplasts, that they originated from free-living prokaryotic cells that were engulfed by an ancestral eukaryotic cell. So an early eukaryotic ancestor, the idea is that it engulfed in oxygen using non-photosynthetic prokaryote, which eventually became an endosymbiont. And an endosymbiont, it is a cell that is living within another cell. And over time, the endosymbiont and the host, it merged into a single organism, which formed the eukaryotic cells with mitochondria. And later, one of those eukaryotic cells, it engulfed a photosynthetic prokaryote, which led to the evolution of chloroplasts containing eukaryotic cells. And there is evidence to support this theory, such as the mitochondria and chloroplasts having two membranes, unlike organelles from the endomembrane system. They contain ribosomes and circular DNA, which is similar to prokaryotic cells. And their DNA, it directs a protein synthesis, which are produced on their own ribosomes. And both organelles, they're going to grow and reproduce independently inside of the cell and function as a semi-autonomous organelle. So mitochondria, they are present in almost all eukaryotic cells, and they serve as the primary site of cellular respiration by converting chemical energy into ATP. And some cells, they might contain one single large mitochondrion, or it can contain hundreds to thousands of smaller mitochondria. And the number of mitochondria found in a cell, it's going to be directly related to the aerobic metabolic activity of that cell. So our mitochondria, they have this smooth outer membrane and a really highly folded inner membrane, which is what we see here with all these foldings. And those inner membranes, it's going to form a structure called cristae. And the inner membrane, it's going to create two distinct internal compartments. It's going to create the intermembrane space, which is the narrow region between the inner and the outer membranes. So like this space right here. And it's also going to create the mitochondrial matrix. And the mitochondrial matrix, it is a fluid-filled space that contains DNA, ribosomes, and enzymes, like that. And our mitochondria, they're really important for cellular respiration. And the key metabolic reactions of respiration, it's going to occur inside of that mitochondrial matrix, and it's going to be catalyzed by enzymes. So our proteins that are involved in ATP production, including such as ATP synthase, they're going to be embedded inside of that inner membrane. And the function of the cristae is to increase the surface area so that it can enhance energy production and efficiency. And our mitochondria, they are usually 1 to 10 micrometers in length. They are dynamic organelles, and they are capable of moving, changing shape, fusing, and dividing as the cell needs. So chloroplasts, they are the site of photosynthesis. They contain something called chlorophyll. They contain molecules and enzymes that are just really essential for the production of sugar. They measure about 3 to 6 micrometers in diameter. They're found in leaves and other green parts of plants, and even algae, right? So they're found in plants and algae. And the chloroplast, it has a structure. It's actually enclosed by an envelope of two membranes, and then it has this really, really narrow intermembrane space that separates them from the cytosol. So when we're looking inside of the innermost membrane, we have a fluid-filled space that's called the stroma. So this like lightish greenish color, that is the stroma. And the stroma, it's going to contain chloroplast's DNA, ribosomes, and enzymes. And inside of the stroma, suspended, are thylakoids. And thylakoids, they are flattened membranous sacs, and they're extremely involved in capturing light. And in some regions, our thylakoids, they are stacked into grana, which is what one of these entire things is. And the purpose of it is to increase the surface area for photosynthesis. So our chloroplasts, they're divided into those three components, into intermembrane space, stroma, and their thylakoid membranes. So our chloroplasts, they are also extremely dynamic. And like mitochondria, they are dynamic and mobile. They move along cytoskeletal tracts inside of the cell, and it belongs to the plastid family. So peroxisomes, they are single membrane bound organelles, and they are going to contain enzymes that are responsible for oxidation reactions. So these enzymes, they're going to transfer hydrogen atoms to various molecules, from various molecules to oxygen, and what it's going to do is it's going to form hydrogen peroxide as a byproduct. And these peroxisomes, they have a few functions. One of their functions is fatty acid breakdown. And fatty acid breakdown, the peroxisomes, it's going to break them down into smaller molecules that are going to be transported to the mitochondria to serve as fuel for cellular respiration. It serves in detoxification. So in our liver cells, peroxisomes, they're going to detoxify alcohol and also other harmful substances by transferring hydrogen atoms to oxygen. And our peroxisomes, they're able to manage hydrogen peroxide. They're able to neutralize hydrogen peroxide. So hydrogen peroxide in itself is toxic. And our peroxisomes, they contain an enzyme that's called catalase. And catalase is going to convert hydrogen peroxide into water, which is going to prevent cellular damage. And we even have these specialized peroxisomes called glyozysomes. And these specialized peroxisomes, they're going to be found in plant seeds. They're going to convert fatty acids in seeds into sugars, which is going to provide them with energy and carbon until that little seedling can perform photosynthesis on its own. These peroxisomes, they're only found in plant cells. The glyozysomes are only in plant cells, but peroxisomes, they're found in both plant and animal cells. And peroxisomes, they grow by incorporating proteins and lipids that come from the cytosol and the endoplasmic reticulum and even the cytosome itself. And they're going to divide by fission. So once they reach a certain size, they're going to undergo fission. And their ability to do fission, it's also suggesting the possibility of an endosymbiotic origin as well. But that theory, it's really still debated at this time. So the cytoskeleton, it is a fibrous network of proteins that extends through the cytoplasm, which is going to provide structural support and maintenance of the cell shape. And the cytoskeleton is especially important in our animal cells because our animal cells do not have cell walls, okay? Animal cells do not have cell walls. So our cytoskeleton, it kind of acts as an anchor for a variety of organelles and even cytosolic enzymes. It also is going to play a really important role in motility, in cell motility, and it's going to enable cell movement and intracellular transport of organelles and molecules. And the cytoskeleton, it is highly dynamic, just meaning that it can be disassembled and reassembled in different regions of the cell, which is going to allow for cell shape changes. So the cytoskeleton is composed of 3 types of protein filaments, and every single filament, it has a distinct function and structural characteristic, right? So our microtubules, they are composed of tubulin. They are the thickest component of the cytoskeleton, and it's going to provide structural support. It's going to help in cell division, and it's going to serve as tracks for organelle movement. And our microtubules, they're also going to form structures like cilia and flagella, which are important for cell motility. Up next, we have our microfilaments, and they are made up of actin subunits, which is why they get the name of actin filaments as well. They are the thinnest component of the cytoskeleton, and they're really important for cell shape, muscle contraction, and movement. And they also play a really important role in intracellular transport and even in cytokinesis, which is another form of cell division. And lastly, we have intermediate filaments. And our intermediate filaments, they're made up of a variety of fibrous proteins, which include keratin. And the diameter of these intermediate filaments, it's between microtubules and microfilaments. These intermediate filaments are going to provide mechanical support, and it's going to help maintain cell shape. It's more stable and permanent than microtubules and microfilaments. So the cytoskeleton is going to interact with motor proteins, which are going to facilitate cell movement and intracellular transport. Motor proteins, they are specialized proteins that transport cellular material along the cytoskeleton. So we have whole cell movement, right? We have whole cell movement. And it's going to include cytoskeletal elements, motor proteins, and the plasma membrane components working together to move that entire cell along external fibers. Cilia and flagella, they're going to move due to motor proteins gripping microtubules and sliding them past one another. And muscle contractions, they're going to occur through a similar mechanism, which is going to involve actin filaments and myosin motor proteins. So, when we're looking at the figure, what we see is a vesicle right here. And that vesicle, it's the cargo, it's the material that has to be transported inside of the cell. And the vesicle, it has these specific receptors, which are the motor protein receptors. And those receptors are where our motor proteins are going to attach to. And then we have our motor proteins. And our motor proteins, they are powered by ATP. The motor proteins, they bind to both the vesicle here and to the cytoskeletal filament. It's going to move along that filament in a walking motion by using ATP as its energy source. And then we have our microtubule of the cytoskeleton. It's just a structural component of that cytoskeleton that's functioning as that track for that vesicular movement. So basically, we have this motor protein that's going to bind to the vesicle and move along the microtubule by using ATP as energy. This movement, it's often compared to walking as if motor proteins have two legs that can take alternating steps. This mechanism, it's going to enable the transport of vesicles, of organelles, and of molecules to different locations inside of the cell. And it's extremely important for intracellular transport, including release of neurotransmitters, organelle positioning, and even the endomembrane system's function. So microtubules, we already said that they are made of tubulin, and they are globular proteins. And our tubulins, it's a dimer. And what that means is that it's going to consist of two subunits, which is alpha tubulin and beta tubulin. Our microtubules, they are dynamic. They are dynamic because they can lengthen or shorten by the removal or the addition of tubular dimers. So this characteristic makes it dynamic. And that plus end of the microtubule, it can accumulate or release dimers faster than the minus end. So our microtubules, they're really important in cellular shape. They're going to provide mechanical support and mechanical support and maintain that cell structure. It's going to work in organelle transport. It's going to serve as tracks for motor proteins that are going to move these vesicles and organelles. And they're important for separating chromosomes during cell division. They're going to form something called mitotic spindles, and that's going to help divide the chromosomes during cell division. So, in a lot of animal cells, our microtubules, they're going to grow out of something called a centrosome. And that centrosome is going to be near the nucleus. And these microtubules, they're really good at resisting compression to the cell. So when the centrosome, when it's a pair of centrioles, like we see here, these two red structures, when it's a pair of centrioles, they're going to each have these 9 triplets of microtubules that are arranged in a ring. And before an animal cell divides, the centrioles, they're going to replicate. And even though our centrosomes with centrioles, they might help to organize microtubule assembly inside of our animal cells, they aren't essential for this function. And in fact, fungi and most plant cells, they lack centrosomes with centrioles, but they have these really well organized microtubules. So cilia and flagella, they are these structures that allow our cells to move. And the cilia and flagella, they are special microtubule structures that are going to help our cells to move by 2 different ways. So first, they're going to extend from the cell, and it's going to extend from a tissue layer, and then they're going to move, they're going to beat in order to move the fluid across the tissue surface. And the way that it's going to happen is that we have cilia, and our cilia, it's going to move back and forth. So it's going to move back and forth, and it's going to move that fluid across the tissue surface. And then we're going to have flagella. And flagella, it moves like a propeller. It moves like this. How a propeller moves. And it's like an undulated wave-like motion that's going to help our cells move. So our cilia and flagella, they are different, okay? But they do have a unifying feature, we can say. So even though they're different, they do share this common structure, which is their microtubules. And their microtubules, they follow this 9+2 pattern because they're going to have 2 central microtubules, and then they're going to have 9 radial spokes. 1, 2, 3, 4, 5, 6, 7, 8, and 9. And this 9+2 pattern, it's found in nearly all of the eukaryotic flagella and motile cilia. And the cilia or the flagella is going to be anchored in a cell by its basal body. And that basal body, its structure is identical to that of a centriole. And in a lot of animals, including humans, that basal body of the fertilizing sperm's flagellum is going to enter the egg and become a centriole. So the motor protein dynein, it's really powerful. It's going to power the cilium and the flagella, and it's going to use ATP as its energy source. The dynein is responsible for the bending movement of the organelle as they perform these complex cycles of movement, which is caused by changes in the shape of the protein. So dynein proteins, they're going to help cilia and flagella bend by walking along microtubules. So dynein, it has 2 feet, and one is going to stay attached while the other one steps forward. And if the feet were not restrained, those microtubules, they were going to just slide right past each other, which was going to make those structures longer instead of bending. So for bending, dynein, it needs something to pull against, like how our leg muscles pull on bones to move. So we have these radial spokes and things called cross-linking patterns, proteins that are going to hold those microtubules in place. And that's going to prevent the excess sliding. And instead, it's going to cause curving that's going to allow that cilia and flagella to bend and move. So now we're going to talk about our microfilaments. Our microfilaments, they are known as actin filaments as well. They are about 7 nanometers in diameter, and they're present in all eukaryotic cells. Each microfilament, it's built like a twisted double chain of actin subunits. And these microfilaments, they can form structural networks because of their ability to branch. And the structural role of these microfilaments in the cytoskeleton is to bear tension and resist pulling forces within the cell. Our cortical microfilaments, those are a 3-dimensional network that is found inside of the plasma membrane, and it's going to help to support the cell's shape by giving the cell cortex a semi-solid consistency, like a gel. So microfilaments, they help with movement a lot, especially in muscle cells. Our muscle cells, they actually have actin filaments that are parallel to each other. And in between those parallel actin filaments, they have something called myosin filaments. And myosin, it moves along actin, which is what makes the muscle contract. So actin, myosin interactions, it also helps in other cellular movements. So for example, during cell division, we have a ring of these microfilaments that contract and then they form a cleavage furrow and it splits the cell. In amoeboid movement, that cell extends pseudopodia, right? Pseudopodia, they are cellular temporary projections, and it extends this by assembling and contracting on actin filaments. And myosin, it's going to pull on actin at the cell's back and push fluid forward into that pseudopodium. And that's what's going to cause it to extend. So some animal cells, like for example, white blood cells, they also use this mechanism to move, to crawl. And this figure is showing amoeboid movement. So the back part of this amoeba, it has this gel-like state fluid called sol, which is going to allow that cytoplasm to move forward. And then this front tip, it extends as that cytoplasm moves backwards. And the front is going to change into that sol, which is a gel, and that's going to help the amoeba move forward. And this cycle is going to repeat. It's going to push the amoeba in the direction of the movement. And this entire process, it's controlled by actin and myosin filaments. So our intermediate filaments, they are about 8 to 12 nanometers. Okay, they're about 8 to 12 nanometers in diameter, which makes them thicker than our microfilaments, but thinner than our microtubules. Their composition is that they're made up of different proteins, such as keratin. They are going to bear tension and provide structural support, and they are the most permanent cytoskeleton element. Even when a cell dies, these filaments, they have the ability to persist and remain. And these intermediate filaments, they're going to hold organelles in place. It's going to reinforce that cellular shape, and they're even going to form a supportive cage kind of around the nucleus. And that's going to help to build that nuclear lamina. And if you remember, nuclear lamina is the inner layer of that nuclear envelope. So we can think of intermediate filaments like the scaffolding of the cell. It's going to help it maintain shape and stability. So the extracellular matrix, it is found outside of cells. It is like in between our cells, we find our extracellular matrix. It is a network of materials that is found between cells. And majority of cells, they're going to synthesize and secrete materials to the outside of the cell. And the inside and the outside of the extracellular matrix, it's connected through cellular membrane proteins, which are going to link that extracellular matrix to that cytoskeleton. Like these integrins, those are those proteins that are going to link the inside and the outside. So in plants, their cell wall, its function is to protect that cell, and it's supposed to maintain the shape. It's going to prevent too much water intake, and it's going to support the plant against gravity as well. It's made up of cellulose microfibrils, which are produced by cellulose synthase. It's going to be embedded in a matrix of proteins and polysaccharides, sort of like a reinforced concrete. And these plant cell walls, they're going to have different types. So they're going to have a primary cell wall, and that primary cell wall, it's like this really thin and flexible wall that's going to help our young cells grow. We have the middle lamella. The middle lamella is this thin and sticky layer that has these things called pectins, and it's going to hold plant cells together. And then we have a secondary cell wall. And the secondary cell wall, it's stronger. It's going to form after growth stops. So for example, wood is mostly made up of secondary walls. And some other features of our plant cell walls is that our microtubules, they're going to guide cellulose deposition, which is going to affect cell growth. So our animal cells, they do not have a cell wall, but they do have this really, really elaborate extracellular matrix. And the primary constituents of this extracellular matrix, they are glycoproteins, so proteins that have carbohydrates. And it includes things especially like collagen fibers, which are going to be embedded into a network of proteoglycans. collagens. And collagen, it accounts for more than half of the total protein that we have in the human body. And a proteoglycan molecule, it consists of these really small core proteins with carbohydrate chains that are covalently attached, and it makes up about 95% of a carbohydrate. And then we also have large proteoglycan complexes that can form when hundreds of proteoglycans become non-covalently attached to one single long polysaccharide molecule. So in many cells, fibronectins, which are found in the extracellular matrix, they're going to connect to integrins, which is what we see here. This is the fibronectin connected to this integrin protein. And cell surface receptor proteins, they're going to span the membrane and they're going to bind on their cytoplasmic side. This is the cytoplasmic side. So they're going to bind onto the cytoplasmic side to proteins which are attached to microfilaments of that cytoskeleton. And these interconnections between the extracellular matrix to the cytoskeleton via this fibronectin integrin link, it's going to permit that integration of changes inside and outside the cell. The extracellular matrix, it has the ability to regulate cell behavior, and there's mechanical signaling that's involved between fibronectin, integrins, and microfilaments of the cytoskeleton. So if we have changes within the cytoskeleton, it can actually trigger chemical signaling pathways inside of the cell, which is going to lead to a change in that set of proteins that's being produced by the cell and then change that cell's function. So we have something that are called cellular junctions, and cellular junctions, they're these specialized structures that are going to connect neighboring cells and actually facilitate communications between them. So like our neighboring cells in our tissues, our organ systems, our organs, normally they connect and they interact and communicate through direct and physical contact. And the point of these cell junctions is to maintain tissue integrity. It's to regulate that cellular communication, and it's to control the passage of substances between our cells. And we're going to have different types of intercellular links. It's going to include tight junctions, desmosomes, and gap junctions. And all three of these intercellular junctions, they are really common, especially in epithelial tissue, which is going to line the external and internal surfaces of the body. So in tight junctions, what we see is that membranes of adjacent cells, they're fused together. It's going to form kind of like a continuous belt around cells, and that's going to stop the leakage of extracellular fluid. See what I mean by our tight junctions? We're going to have desmosomes. These are our desmosomes. We call our desmosomes anchoring junctions as well. They're going to fasten cells together into these like really strong sheets, and intermediate filaments, they're actually of keratin, which is reinforced by desmosomes. And we're also going to have these gap junctions, which are our communicating junctions. It's going to provide cytoplasmic channels between our adjacent cells. It's a special membrane protein that surrounds these pores, and ions, sugars, and amino acids, and other small molecules. They have the ability to pass. And in our plant cells, we have something similar to a gap junction. It's called plasmodesmata. It's found in plant cells, and it's like perforations that is going to allow for cytosol to pass between our cells.